I am from Government Degree College Autonomous Rajamahendravaram, Department of Biotechnology. Myself V. Sirisha from BSc Honors Biotechnology, second year, third sem. Today my seminar topic is somatic hybridization. What is somatic hybridization? Somatic hybridization is the technique of hybrid production through the fusion of isolated somatic protoplast under in vitro conditions. This somatic hybridization involves in 5 steps. They are isolation of protoplast, fusion of the protoplast of desired species, selection of somatic hybrid cells, culture of the hybrid cells, regeneration of the hybrid plants. In this isolation of protoplast involves in some methods. First method is mechanical method. In this method, plant tissues under 1.0 molar sucrose. Then cell plasmolysis. Later, microscope absorption of cells. Cut only the cell wall with knife. Then cell releases the protoplast. Then we collect the protoplast. Another method is enzymatic method. Pectinase mainly degrades the middle lamella, while cellulase and hemicellulase are required to dissect the cellulosic and hemicellulosic components of the cell wall respectively. In this enzymatic method involves in 2 methods. Simultaneous is one step method. In this method, the tissues is subjected to a mixture of enzymes in a one-step reaction, which includes both macroenzymes or pectinase and cellulase, and sequential or second step method. The tissues is first treated with a macroenzyme or pectinase enzyme, which separates the cells by degrading the middle lamellae. Cellulase, which releases the protoplast. In this somatic hybridization, second step is protoplast fusion. It is the technique in which 2 or more protoplasts are fused into a single cell protoplast. Methods of protoplast fusion. First one, spontaneous fusion. Protoplasts fuse spontaneously during isolation process, mainly due to physical contact among similar parental protoplasts. 2 types of spontaneous fusions. Intraspecific produce homokaryons. Intergeneric have no importance. Another one, induced fusion. In induced fusion, they mechanical fusion. In this, the isolated protoplasts are brought into intimate physical contact mechanically using micromanipulator. In this induced fusion, second type, chemical fusion. In this chemical fusion, first one, PEG treatment. Isolation protoplasts in culture media are mixed with equal volumes of 28 to 56% PEG in a tube. Tube is shaken and then allowed to select unselected protoplasts are washed several times with culture medium. During which fusion occurs, this polymer binds to the liquid membrane of cells and this induces fusion. Second one is NaNO3 treatment. Isolated protoplast exposed to a mixture of 5.5% NaNO in 10% of sucrose solution. Incubate carried out for 5 minutes at 35 degrees Celsius, followed by the centrifugation. And next one, treatment with calcium ions at high pH. The method consists of incubating protoplast in a solution of 0.4 molar mannitol containing 0.05 molar calcium chloride at pH 10.5 at 37 degrees Celsius for 30 to 40 minutes. In this somatic hybridization, third step is selection of somatic hybrid cells. In this, first one is complementation. In this method, complementation of genetic or metabolic deficiencies of two fusion parents are utilized to select hybrid components. When protoplast of two parents each carrying non-allelic genetic or metabolic defects are fused, it reconstitutes a viable hybrid cells of wild type in which both defects are mutually removed. Merkels and Labrid 1974 demonstrated this to wild type green Cali and plant after fusion of protoplast from two chlorophyll deficient varieties of tobacco. In this selection of somatic hybrid cells, second method is chlorophyll deficiency complementation. Merkels and Labrid 1974 first used genetic complementation to isolate green somatic hybrid following fusion of two distal homozygous receive albino mutations of Nicotiana tabacum. Third method is use of visual characteristics. Use of morphological distant cells. Fusion of protoplasts containing green chloroplast with colorless cells and cultured protoplast contains distant starch granules due to growth on sucrose supplements medium. Fusion products can be seen immediately with one of containing chloroplast and other of with distant starch granules. In this somatic hybridization, fourth step is culture of the hybrid cells. Hybrid cells are cultured on suitable medium provided with the appropriate culture conditions. And fifth step is regeneration of hybrid plants. Plants are included to regenerate from hybrid cells. Applications of somatic hybridization. Novel approach for increasing genetic viability. Production of novel interspecific and intergenic hybrids. Example, tomato. Production of fertile diploids and polyploids. Transfer gene for diseases resistance, abiotic stress resistance, herbicide resistance, and many other quality characters. The production of unique nuclear cytoplasmic combination. Disadvantages of somatic hybridization. Poor regenerations of hybrid plants, non-viability of fused products, not successful in all plants, production of unfavorable hybrids, lack of an efficient method for selection of hybrids. Thank you for watching this video.